There is a skeleton in New Mexico that should not exist. Not in the metaphorical sense, not as a rhetorical flourish. In the most literal, stratigraphically precise, radiometrically verified sense available to modern geology, the bones recovered from a sandstone quarry in the San Juan Basin represent something that the entire framework of vertebrate paleontology insists cannot be real, and yet the rock does not lie. The iridium is exactly where it should be. The zircon crystals are exactly where they should be. The theropod skeleton is exactly 1.5 million years above both of them, embedded in sediment that was being laid down while the first post-extinction mammals were already diversifying, while ferns were colonizing a world still recovering from the most catastrophic ecological collapse in the history of complex animal life. The bones are there. They are articulated, they are unambiguous, and they belong to a non-avian dinosaur. Stay with me here, because the magnitude of what that stratigraphic position actually means requires a moment to absorb fully. The Cretaceous-Paleogene boundary is not a soft or contested geological marker. It is one of the most precisely identified and globally replicated chronological signatures in the entire sedimentary record of this planet. At sites distributed across every continent and in deep-sea cores drawn from every major ocean basin, the same chemical fingerprint appears at the same moment in geological time. A thin, laterally continuous layer of clay enriched with iridium at concentrations roughly 30 times above normal crustal background values, accompanied by microscopic spherules of once molten rock, grains of quartz bearing the characteristic planar deformation, features produced only by hypervelocity impact, and in many sites, a thin veneer of soot consistent with global wildfires of extraordinary scale. This layer marks the moment 66 million years ago, when a bolide approximately 10 to 12 kilometers in diameter struck the shallow carbonate shelf of what is now the Yucatan Peninsula of Mexico, releasing an energy equivalent that most estimates place in the range of 1 billion times that of the atomic bomb dropped on Hiroshima. The iridium layer is the calling card of that impact. Below it, non-avian dinosaurs. Above it, none. None, that is, until the San Juan Basin. The specimen came out of the Nacimiento Formation, which sits conformably above the Hell Creek equivalent lithology at this particular site. This matters enormously. The stratigraphic column here is not disrupted. There is no fault cutting across the section, no unconformity suggesting a gap in deposition, no angular relationship between the lower and upper beds that might imply erosion and redeposition. The sequence reads cleanly and continuously from uppermost Cretaceous mudstone through the boundary clay and upward into Paleocene floodplain deposits, and the theropod is embedded in those uppermost Paleocene sediments with the same orientation and spatial coherence that implies it died where it lay rather than having been transported, reworked or redeposited from an older source. Radiometric dating is where the case becomes truly unassailable. Volcanic ash beds, the thin white laminae of airfall tephra that geologists treat as absolute time markers because their zircon crystals lock uranium and lead ratios at the precise moment of crystallization, bracket the fossil horizon from both above and below. Uranium-led zircon analysis of those ash beds produces ages that are internally consistent and analytically reproducible. The lower ash bed dates to approximately 65.8 million years ago. The upper ash bed dates to approximately 65.4 million years ago. The fossil horizon sits between them. The animal, by every available geochronological method, was alive approximately 65.5 million years ago. The globally recognized iridium anomaly marking the Chicxulub impact and the accepted end of the non-avian dinosaur lineage falls at 66 million years. The arithmetic here is not complicated. 1.5 million years separate the last accepted moment of non-avian dinosaur existence from the sediment that holds these bones. Now consider what that interval actually represents in biological time. 1.5 million years is not a rounding error. It is not a measurement uncertainty. 1.5 million years is approximately the entire span of time that separates the earliest members of the genus Homo from the present moment. It is long enough for substantial evolutionary divergence. It is long enough for ecosystems to collapse and partially recover. It is long enough for climates to shift through multiple cycles of warming and cooling. Whatever organism left these bones behind was not a creature that barely scraped through the KPG boundary by a geological whisker. It was an animal whose lineage had been living, feeding, and presumably reproducing in a post-extinction world for a duration that dwarfs the entirety of recognizable human prehistory. Biostratigraphic cross-referencing reinforces what the radiometric data establishes. The matrix immediately surrounding the skeleton contains pollen assemblages dominated by fern spores, specifically the characteristic proliferation of opportunistic ferns that colonized devastated landscapes in the immediate aftermath of the KPG event, and which are recognized globally as one of the most reliable biological signatures of the post-impact recovery interval. Alongside these fern spores are early angiosperm pollen types and fungal spore assemblages that are entirely consistent with the disturbed, low-diversity, decomposer-heavy ecology that characterized Paleocene terrestrial environments in the western interior of North America. There are also mammalian microfossils in the matrix, specifically the fragmentary teeth and jaw elements of archaic condylarths, a group of early placental mammals that diversified explosively in the ecological vacuum left by the extinction of the dinosaurs. These are unambiguously Paleocene organisms. They have no Cretaceous antecedents at this site. Their presence in the same matrix as a theropod skeleton is, by every prior assumption of the discipline, impossible. And this is the question that drives everything that follows, not simply how this animal survived the impact, because the fossil record is full of survivors, and the KPG boundary was not a perfect filter. Birds survived, crocodilians survived, turtles and lizards and early mammals survived. The question is far more specific and far more demanding. If every accepted mechanism of the KPG extinction event, the initial thermal pulse of the impact, the global wildfires, the sulfate aerosol cooling, the collapse of photosynthetic productivity, the acid rain, the decade-scale darkness of stratospheric particulate loading, if all of those mechanisms acting in combination were sufficient to eliminate every other lineage of non-avian dinosaur on a global scale, then what precise combination of biological machinery, physiological tolerance, behavioral flexibility, and ecological circumstance allowed this particular lineage to persist through all of it, and persist not for decades or centuries but for 1.5 million years, and then, far more unsettling, what finally ended them. Because here is what the fossil record insists must be true. This animal survived the unsurvivable. It outlasted the impact winter. It outlasted the ecological collapse. It adapted demonstrably and measurably to conditions that killed everything else, and then something killed it anyway. The bone histology tells part of that story. The endocranial anatomy tells another part. The isotopic chemistry locked inside the mineral lattice of its teeth tells the most disturbing part of all. But to understand any of what that skeleton eventually reveals about how it lived and how it died, you have to start by understanding exactly how it was found, and what the rock itself says about whether this animal truly existed where and when the stratigraphy insists it did. The sedimentary column at the San Juan Basin site is where that answer begins. The question of whether a fossil is truly where the rock says it is sounds almost philosophical, until you are standing in front of a stratigraphic section and walking it metre by metre with a hand lens and a measuring rod. Then it becomes entirely physical. The sediment either grades continuously from one unit to the next, or it does not. The contacts are either conformable, or they are not. The rock tells you, directly and without ambiguity, whether what you are looking at is a continuous depositional sequence, or a sequence that has been interrupted, inverted, disturbed, or chemically contaminated. At the San Juan Basin site, the excavation team spent the better part of 2 field seasons doing exactly this work before a single interpretive claim about the theropod's age was committed to print. What they documented is a stratigraphic column that reads like a textbook of the KPG transition, except that it continues past the page where every other textbook stops. The lowest exposed beds at the excavation face are fine-grained floodplain mudstones and overbank siltstones characteristic of the Hell Creek equivalent lithology that dominates uppermost Cretaceous terrestrial deposits across the western interior of North America. These sediments are oxidized, brick red to purple in places, and contain root traces and pedogenic carbonate nodules consistent with well-drained, seasonally dry floodplain environments. The fauna preserved within them, documented through systematic screenwashing of the surrounding matrix, is classically late Maastrichtian in character. Fragmentary hadrosaur and ceratopsian bone chips, lizard osteoderms, and the teeth of multituberculate mammals whose lineage stretches deep into the Cretaceous. This is the familiar world. This is the world that ends. Moving upward, the transition is abrupt in the way that only geological catastrophe produces. Within a vertical interval of less than 3 centimeters, the depositional character of the sediment changes completely. The brick-red mudstone gives way to a pale, waxy, clay-rich layer, with a distinctive blue-grey tone that experienced stratigraphers recognize immediately because they have seen its equivalent on every inhabited continent. This is the boundary clay. Geochemical analysis of this layer at the San Juan site produces the expected result: iridium concentrations elevated to roughly 32 times the crustal background value measured in the Cretaceous mudstones immediately below. Alongside the iridium enrichment, the team identifies shocked quartz grains bearing multiple intersecting sets of planar deformation features, oriented at angles consistent with the extremely high pressures produced by hypervelocity impact rather than by volcanic processes or metamorphism, and centimetre-scale spherules of recrystallized glass, with compositions indicating derivation from the vaporization and rapid cooling of continental carbonate and silicate target rock. There is also a thin carbonaceous band within the upper portion of the clay layer that geochemical analysis identifies as soot with a polycyclic aromatic hydrocarbon signature consistent with combustion products from large-scale wildfires rather than from volcanic emissions or normal organic diagenesis. This is the impact horizon. Every marker is present, every marker is in the expected relative position, and every marker is geochemically consistent with its counterparts at dozens of globally distributed KPG boundary sites. Below this layer, Cretaceous organisms, above it a different world entirely, world. And the world above it is exactly what the Paleocene looks like. Immediately overlying the boundary clay, the Nacimiento Formation begins in earnest. Lighter coloured, more poorly sorted floodplain channel sands and siltstones with a completely different pedogenic character. The root trace morphologies change, reflecting different plant communities. The sediment colour shifts toward buff and olive tones consistent with waterlogged, reducing conditions rather than the oxidising floodplain soils below. Pollen extracted from these overlying beds tells the story with exceptional clarity. The dominant signal in the first several metres above the boundary is the fern spike, specifically an overwhelming proliferation of polypodiaceous fern spores that in equivalent sections worldwide represents the opportunistic colonisation of devastated landscapes by pioneer vegetation tolerant of disturbed, nutrient-stripped substrates. The diversity of pollen types is dramatically lower than in the Cretaceous beds below, and the composition has shifted entirely. None of the characteristic late Maastrichtian gymnosperm and angiosperm pollen types that were abundant below the boundary layer appear above it in meaningful quantities. The community has been reset. Higher in the section, as the column approaches the fossil horizon, the pollen record begins to show the first tentative signs of ecological recovery. Angiosperm diversity creeps upward. Fungal spore abundance, which spikes dramatically in the earliest Paleocene layers as decomposer communities exploit the enormous biomass of dead and dying organisms, begins to taper off. The ecosystem is reorganizing slowly and incompletely around new ecological architectures, and it is within this recovering, botanically impoverished, decomposer-heavy landscape that the theropod skeleton sits. The taphonomy of the specimen itself provides the most direct argument against reworking or redeposition. The skeleton is articulated. Not partially articulated, not with bones in approximate anatomical proximity, but genuinely articulated in the sense that means the animal decomposed in place, with the connective tissues holding the skeleton together long enough for burial to preserve the spatial relationships between elements. The cervical vertebrae maintain their natural curvature. The hind limb elements preserve the stacking and angular relationships of the knee joint. Several dorsal ribs are still in contact with their respective vertebral facets. This is a death assemblage, not a transport assemblage. Hydraulic sorting would have separated the bones by density and surface area, clustering the compact distal elements together and dispersing the larger flat elements preferentially. Bioturbation would have scattered elements randomly and introduced vertical displacement. Neither signature is present. The animal lies as it fell. The surrounding matrix, collected systematically from the immediate vicinity of the skeleton and processed through fine mesh screen washing, yields a microfossil assemblage that closes any remaining interpretive door. The teeth recovered from the matrix belong to archaic condylarths, small-bodied early Paleocene mammals whose first appearances in the North American record post-date the KPG boundary by a measurable stratigraphic interval. These are not Cretaceous mammals. Their dental morphology places them unambiguously within post-boundary diversification lineages that have no counterparts in the Maastrichtian fauna below. Their presence in the matrix containing the theropod means that either the mammal teeth were introduced into this sediment from a younger source, which the unbroken and conformable stratigraphy makes geologically implausible, or the theropod and those mammals inhabited the same landscape at the same time. Here is where the first serious methodological challenge arrives, and it is one that the research team could not simply dismiss. A competing group of geochemists reviewing the published boundary layer data from the San Juan site raised the possibility that the iridium enrichment in the boundary clay at this specific location was not a primary depositional signal, but a secondary geochemical concentration produced by hydrothermal fluid migration through the section after burial. The mechanism they proposed is not without precedent. Iridium, like several other platinum group elements, can be mobilized under reducing conditions by low-temperature hydrothermal fluids and reconcentrated along chemical gradients, potentially producing an apparent iridium anomaly at a stratigraphic position that does not correspond to the actual depositional age of the impact fallout. If the iridium layer at this site had migrated upward from its original depositional position by even a small vertical distance, the apparent gap between the impact horizon and the theropod could be an artifact of post-depositional geochemistry rather than a genuine representation of elapsed time. This objection deserved serious engagement rather than dismissal, and the response required going back to the rock with a more detailed geochemical toolkit. The critical test involves examining not just the iridium concentration, but the entire suite of platinum group element ratios preserved in the boundary clay. Hydrothermal remobilization does not move all platinum group elements with equal efficiency because their geochemical behaviors under aqueous conditions differ significantly. Primary impact fallout produces a characteristic ratio of iridium to osmium to ruthenium to platinum that reflects the composition of chondritic meteoritic material. If the iridium anomaly at this site resulted from secondary concentration, the platinum group element ratios should be fractionated, with some elements preferentially enriched or depleted relative to the chondritic pattern. The measured ratios at the San Juan boundary clay are indistinguishable from primary impact fallout signatures at reference sites globally. Furthermore, the shocked quartz grains and glass spherules documented in the boundary layer cannot be mobilized or redistributed by hydrothermal fluids. They are physical particles, and their presence within the clay layer at this site means that at minimum the physical matrix of the boundary layer is in its original depositional position, even if one were to accept the geochemical redistribution hypothesis, which the platinum group element data does not support. The boundary layer is real, it is primary. The gap between it and the theropod is real-time, not geochemical artifact. But confirming that the animal existed where and when the stratigraphy says it did is only the beginning of the problem. Because the bones themselves, once the stratigraphic case was secured, began telling a story that the team had not anticipated finding. The bones themselves are the argument. Everything established about the stratigraphic position, every geochemical test, every radiometric confirmation, all of it tells you when and where this animal existed, but the skeleton tells you what it had become. And what the San Juan theropod had become is something that has no direct counterpart anywhere in the 160 million year history of non-avian dinosaurs. Start with the most immediately visible feature, the one that struck the excavation team before a single histological section had been cut: the cortical bone. In virtually every large coelurosaurian theropod recovered from Cretaceous deposits, the long bones of the hind limb display a characteristic architecture that reflects the evolutionary pressures of active, energetically demanding locomotion. The cortex is relatively thin, the medullary cavity is spacious, and the bone walls are perforated by a dense network of vascular channels that supplied blood to the metabolically active tissue of a fast-growing high-energy animal. In many advanced coelurosaurs, the axial skeleton and limb girdle elements are further lightened through pneumatization. The invasion of air sac diverticula from the respiratory system into the bone leaving hollow chambers within the vertebrae and pelvic elements that reduce overall skeletal mass without compromising structural integrity. This pneumatized, vascularized, lightweight skeletal architecture is so consistently characteristic of the Coelurosaurian body plan that it functions almost as a taxonomic identifier. The San Juan specimen's cortical bone is the precise opposite of all of that. The femur, the most completely preserved long element in the skeleton, has a cortical wall thickness that exceeds what would be predicted for an animal of its estimated body mass by a factor of roughly 2.5. The medullary cavity is dramatically reduced, partially occluded by endostele bone infilling that has progressively narrowed the internal space over what the growth record suggests were multiple years of deposition. The vascular porosity of the cortex itself, measured through quantitative histomorphometry on thin sections cut transverse to the long axis of the bone, is reduced to approximately 1/3 of the values recorded in comparable Cretaceous tyrannosauroids of similar estimated body size. What you are looking at if you hold one of these thin sections up to a light source is dense, almost marble-like bone tissue with a compactness that calls to mind the skeletal architecture of something like a manatee or a mosasaur rather than a terrestrial cursorial predator. This condition has a formal name, osteosclerosis, and its occurrence in the San Juan specimen is not a pathological accident. Osteosclerosis of this degree and this distribution across the skeleton is documented in the fossil record in specific and well-understood ecological contexts. It appears in secondarily aquatic tetrapods, animals whose lineages return to water from a terrestrial ancestry and whose dense bone provided ballast for buoyancy control in shallow aquatic environments. It appears in large marine reptiles, in some sauropods whose bone histology suggests semi-aquatic habits, and in certain island-evolved herbivores whose reduced locomotor demands apparently relaxed selection against the energetic cost of maintaining denser skeletal tissue. In all of these cases, the functional interpretation is consistent. Osteosclerosis represents a systemic shift in the biomechanical and metabolic priorities of the skeletal system, away from lightweight rapid movement and towards structural mass, thermal inertia, or some combination of physiological functions that benefit from denser, less vascularized tissue. Think about what that means for an animal living through an impact winter. Dense, poorly vascularized bone is bone that conserves heat. It is bone that does not demand the high rates of metabolic turnover associated with rapid growth and active remodeling. It is bone that belongs to an animal running a much slower biological clock than its ancestors did. The growth ring data makes this explicit. Every vertebrate bone grows in seasonal increments, and in temperate or seasonally variable environments, those increments are separated by lines of arrested growth, narrow bands of denser, less vascularized tissue that form during winter or dry season periods of reduced food availability and slower physiological activity. Count the rings, and you can read the years of an individual animal's life. Measure the spacing between rings, and you can estimate the annual rate of somatic growth, which correlates strongly with metabolic rate across a wide range of vertebrate taxa. The femoral growth rings of the San Juan specimen are closely and uniformly spaced in a way that implies an annual somatic growth rate approximately 1/3 of what comparative data from Cretaceous tyrannosauroids of equivalent body size would predict. This animal was growing at a pace consistent with an animal whose metabolic engine was running at dramatically reduced output. Not pathologically reduced, systematically, consistently, chronically reduced in a pattern that extends across the entire preserved growth history of the bone, and that is replicated in the tibial sections with statistically indistinguishable values. But the growth rings contain one more piece of information that pushes this interpretation from interesting to extraordinary. The oxygen isotope ratios measured within the carbonate component of the bone mineral itself, the hydroxyapatite lattice that incorporates oxygen from the animal's body water during bone formation, encode the temperature at which that mineral was precipitated. This is a well-validated paleothermometric tool used extensively in studies of dinosaur physiology and paleoclimate reconstruction. The values from the San Juan femoral cortex indicate bone formation temperatures that are consistently 6 to 9 degrees Celsius below the late Cretaceous regional average, documented from equivalent analyses of Maastrichtian theropod bone at comparative sites. 6 to 9 degrees, that is not measurement noise. That is a sustained, decades-long record of cold embedded in the mineral matrix of the animal's own skeleton. a physiological diary written in crystal chemistry that confirms the bone was forming inside an animal, living through temperatures far below those experienced by any of its Cretaceous predecessors. Now here is where the data starts to ask a question that the bone histology alone cannot answer. The adaptations documented in the cortical architecture are extensive and deeply integrated into the skeletal biology of this animal. Osteosclerosis of this magnitude, growth suppression of this consistency, the thermal signal embedded in the isotopic composition of the bone mineral. These are not superficial modifications. They reflect a fundamentally different physiological program from the one running in any known Cretaceous theropod. And that raises the interpretive question that the research team found themselves returning to repeatedly as the histological dataset accumulated. Were these modifications the product of multi-generational natural selection, acting on a viable breeding population over hundreds of thousands of years, progressively filtering for genotypes that could tolerate cold, hypoxia, and metabolic depression? Or could a single lineage, starting from Cretaceous baseline physiology, achieve histological transformation of this depth through developmental plasticity, the capacity of an individual organism to alter its skeletal architecture in direct response to environmental conditions without requiring genetic change? The distinction matters enormously. If the answer is natural selection operating over a significant span of post-impact generations, then you need a breeding population. You need sufficient individuals surviving in sufficient density across sufficient habitat to avoid genetic erosion through inbreeding and drift across what the stratigraphy suggests was roughly 1.5 million years. That is a cryptic megafaunal presence in the Paleocene that the fossil record until now gave absolutely no indication of containing. If the answer is phenotypic plasticity, a single lineage or even a handful of individuals responding developmentally to environmental stress, then the populational requirements are much more modest, but the physiological implications become even more remarkable, because the degree of skeletal reorganization documented here approaches, and in several metrics exceeds the range of plasticity documented in any living vertebrate taxon subjected to experimental or natural environmental stress. The bone histology cannot adjudicate between these two mechanisms on its own. It can only confirm the magnitude of the transformation and the timescale over which it was sustained. But the skull, which the team had only partially prepared when the femoral histology data came back from the lab, was about to add a completely different dimension to the physiological portrait of this animal. Because the skeleton preserves not just the history of how the body was built, it preserves the architecture of the brain that ran it. The skull came off the matrix in pieces, which is how it usually goes with something this old and this compressed, and the preparation work alone took the better part of 8 months before the endocranial cavity was sufficiently cleared to permit computed tomographic scanning. What the scans revealed when the data finally resolved into 3-dimensional reconstructions was not what anyone on the team had expected to find. And given everything the bone histology had already established about this animal, that is saying something. Let me walk you through the anatomy precisely, because the details here carry the entire argument. The endocast, meaning the natural or artificially reconstructed cast of the space inside the brain case that was once occupied by the brain itself, is the primary tool available for inferring neurological organization in extinct vertebrates. You're not looking at preserved brain tissue. That does not survive fossilization under normal circumstances. What you're looking at is the shape of the cavity the brain occupied, and from that shape, combined with comparative data from living relatives and from the extensive theropod fossil record, you can make defensible inferences about the relative size and organization of different brain regions. The olfactory bulbs leave a characteristic anterior-most impression. The cerebral hemispheres define the middle volume. The optic lobes project laterally. The cerebellar region occupies the posterior portion. Each of these structures, in different species adapted to different sensory and behavioral environments, occupies a characteristic proportion of the total endocranial volume, and those proportions are not arbitrary. They reflect genuine investment in neurological processing capacity that correlates, imperfectly but measurably, with behavioral emphasis on different sensory modalities and cognitive demands. The total endocranial volume of the San Juan specimen, once the reconstruction was complete and the measurements corrected for the taphonomic compression the skull had undergone during burial, comes in at approximately 18% below the value predicted by the scaling relationship between body mass and brain size established from the Cretaceous-Coelurosaurian dataset. 18%. That is not a rounding error in the estimation of body mass. The skeletal dimensions give a reasonably constrained body mass estimate, and even running the calculation at the upper and lower bounds of that estimate, the endocranial volume remains consistently deficient relative to what a Cretaceous animal of comparable size would be expected to carry. This phenomenon has been formally characterized within the research as secondary encephalization reversal, meaning a reduction in relative brain size occurring secondarily within a lineage that had previously achieved a higher encephalization quotient. That pattern has no direct precedent in the theropod fossil record. Nowhere across the documented history of non-avian theropods, from the earliest Triassic forms through the diverse coelurosaurs of the late Cretaceous, is there a documented case of a lineage showing a reduction in relative brain size across an evolutionary transition. The trajectory within theropods is consistently in the other direction. Encephalization increases. The lineage leading to birds represents the endpoint of that trend, with the most highly encephalized non-avian dinosaurs occurring in the latest Cretaceous. The San Juan specimen is running that trend backward, but here is where the anatomy stops being simply anomalous and starts being interpretively coherent. The olfactory bulbs are the critical counterpoint to the overall encephalization reduction. In the endocast of the San Juan specimen, the paired anteriormost projections corresponding to the olfactory bulbs are not reduced. They are massively enlarged. The volumetric proportion of the total endocast occupied by olfactory structures is roughly 3 times the proportional investment documented in comparably sized Cretaceous coelurosaurs. 3 times. To put that in concrete terms, in a typical large Cretaceous coelurosaur, the olfactory region might account for something in the range of 12 to 15% of the total endocranial volume. In the San Juan specimen, the olfactory structures dominate the anterior third of the endocast in a manner that more closely resembles the proportional investment seen in deep burrowing mammals, nocturnal predators, or aquatic vertebrates that navigate in environments where visual information is severely degraded than anything found in a Mesozoic theropod. In living vertebrates, this degree of olfactory hypertrophy is not distributed randomly across ecological guilds. It appears consistently in animals operating in photically impoverished environments where chemical cues carry a disproportionate navigational and foraging load. Moles, deep sea sharks, certain nocturnal squamates, these are animals that have, through selection pressure, redirected neurological investment toward chemosensory processing at the expense of other cognitive capacities because the environment in which they live makes olfactory information more reliable and more immediately actionable than visual or auditory information. In every case, the anatomy is not accidental. It is the physical record of what the animal needed to survive. Now think about the post-impact environment from the perspective of a large terrestrial predator. The stratospheric particulate loading from the Chicxulub event is estimated to have reduced surface irradiance to a fraction of its pre-impact baseline for a period measured in years at minimum, with some climate models suggesting meaningful photosynthetic suppression persisting for a decade or longer. During that interval, and during the extended period of reduced solar input that followed as particulate loading gradually diminished, but did not immediately clear, the visual environment at the surface of the Earth would have been profoundly degraded. Not total darkness, but chronically dim, diffuse, and unreliable as a navigational medium in ways that would have substantially impaired a visual predator's ability to locate and track prey. An animal navigating by smell in that environment would have been operating in conditions that paradoxically played to its reorganized neurological strengths. The collapsed photosynthetic food web would still have generated chemical gradients as organic matter decomposed, and microbial communities processed dead biomass. Those gradients would have been detectable. A predator with tripled olfactory investment and dramatically reduced dependence on visual processing would have been able to find food in conditions that would have left a visually dependent predator effectively blind. The optic lobes confirm this interpretation by showing the expected reciprocal reduction. The lateral projections of the midbrain corresponding to the optic lobes, which in Cretaceous coelurosaurs are robustly developed and reflect the visual acuity and tracking ability that made these animals effective pursuit predators, are substantially compressed in the San Juan endocast. The proportional reduction in optic lobe volume mirrors almost precisely the proportional increase in olfactory investment, as if the neurological reorganization proceeded by a kind of reallocation of existing endocranial space, rather than by any absolute increase in overall brain volume. Whether that is literally what happened at a developmental level is a theoretical question that the endocast alone cannot resolve, but the geometric relationship between the two changes is striking enough to suggest that they were functionally linked. The inner ear tells one more piece of this story. The petrosal bone, the dense bony capsule that houses the sensory structures of the inner ear, including the semicircular canals responsible for detecting angular acceleration and maintaining vestibular equilibrium, was sufficiently well preserved in the San Juan specimen to permit detailed reconstruction of the canal geometry. The semicircular canals in living and extinct vertebrates follow a scaling relationship in which canal radius correlates with the agility and responsiveness of the animal's locomotion. Large, widely spaced canals with generous radii are associated with rapid, precise head movements and high agility locomotion. The canal geometry of the San Juan specimen, compared against the established dataset for large coelurosaurian theropods, falls well below the expected values for an animal of its body size, suggesting locomotor behavior that was substantially slower, more deliberate and less dynamically demanding than what would be inferred for a Cretaceous relative of comparable mass. Slow-moving, olfaction-dependent, neurologically reorganized away from visual processing and toward chemosensory guidance, operating in chronic cold with a metabolic rate far below baseline. And here is the question that the skull now forces onto the investigation with a directness that the bone histology could only imply. What was this animal actually eating? The endocranial and inner ear anatomy describes a forager, not an active predator. The metabolic suppression documented in the growth histology describes an animal with a drastically reduced caloric requirement. But even a reduced caloric requirement is still a requirement, and a large-bodied terrestrial vertebrate in the most energy-impoverished ecosystem the Cenozoic has ever produced still needs to find enough food to survive, grow, and if the population modelling requires it, reproduce. The chemistry of its own teeth holds the answer to that question. The teeth are small. Relative to the animal's estimated body mass, the individual tooth crowns recovered from the jaw matrix are notably gracile, with a zyphodont morphology, meaning laterally compressed, blade-like and serrated along both the mesial and distal carinae, that is ancestrally characteristic of carnivorous theropods across the entire coelurosaurian radiation. If you handed one of these teeth to a vertebrate paleontologist, without any stratigraphic context, and asked them to characterize the animal's diet, the answer would come back almost automatically. Carnivore, active predator. That is what the tooth form says at first glance, and that first glance is wrong. The chemistry tells a completely different story. Stable isotope analysis of biological hard tissues has become one of the most powerful dietary reconstruction tools available to paleontology, precisely because it bypasses morphological assumption and reads the biochemical record of what an animal actually consumed across its lifetime, rather than what its anatomy suggests it was equipped to consume. The fundamental principle rests on the fact that different food sources carry distinct isotopic signatures that become incorporated into the mineral matrix of teeth during formation. And because enamel is metabolically inert once it crystallizes, those signatures are preserved with remarkable fidelity across geological time. Sampling along the growth axis of a tooth at sub-millimeter resolution, tracking compositional change from the earliest formed enamel at the tooth's base to the latest formed tissue at its tip, gives you a temporal record of dietary chemistry across the interval that tooth was growing. Do this across multiple shed teeth recovered from different positions in the jaw, which represent different growth intervals in the animal's life, and you begin to assemble something close to a continuous dietary biography. The delta-13 carbon values from the San Juan specimen's tooth enamel are the first thing that forces a reassessment of the morphology-based carnivore assumption. In a straightforward active predator consuming large vertebrate prey in a functioning photosynthetic ecosystem, delta-13 carbon values in tooth enamel tend to cluster in ranges that reflect the carbon isotopic composition of the plant base, transmitted upward through one or two trophic steps. Those values are relatively predictable and relatively constrained. What the serial sampling of the San Juan teeth produces is a distribution that clusters well below those expected ranges, in a zone that isotope ecologists associate with consumption of organic matter routed through decomposer-based rather than photosynthesis-based food webs. Fungal biomass carries anomalously depleted carbon isotopic signatures because fungi fractionate carbon during the decomposition of organic substrates in ways that plant tissues do not. Detritivorous invertebrates, animals that feed directly on decaying organic material, and the microbial communities processing it, inherit those depleted values. And scavenged megafaunal carcasses, depending on the stage of decomposition at which they were consumed, can carry mixed signals that trend toward depletion relative to freshly killed prey tissue. The San Juan enamel values are consistent with a diet dominated by exactly those sources. Not photosynthesis-derived vertebrate prey, detritus, fungal material, decomposing organic matter, and intermittent scavenging. This is not a predator at the apex of a functioning food web. This is an opportunistic survivor working the margins of a decomposer economy. Stay with the dental microwear data for a moment, because it independently corroborates what the isotopes are saying. Dental microwear texture analysis examines the microscopic abrasion patterns on the occlusal and labial surfaces of teeth using confocal microscopy and scale-sensitive fractal analysis to characterize the mechanical properties of the food items being processed. Hard objects, bone, seeds, beetle carapaces produce a microwear signature dominated by deep pits, large fracture pit complexes, and coarse surface texture. Soft, yielding items, flesh, fungal bodies, invertebrate soft tissue, produce a signature of fine, shallow, densely distributed scratches with low overall surface complexity. The San Juan teeth show the latter pattern comprehensively. Fine scratches dominate. Deep gouges and fracture pits are essentially absent. Whatever this animal was putting in its mouth was soft. It was not crushing bone. It was not processing hard invertebrate prey. It was handling yielding, low-resistance material consistent with the isotopically indicated detritivore fungal dietary base. There is a further detail in the microwear that carries its own physiological implication. The overall rate of dental wear on the San Juan specimens, estimated by comparing the degree of abrasion on teeth of known relative age within the dental replacement sequence, is substantially lower than would be expected for an animal of this body size consuming food at the rates typical of a large-bodied Cretaceous theropod. Theropod teeth are replaced continuously throughout life, but they are also worn continuously through feeding activity, and the balance between replacement rate and wear rate gives you a rough proxy for feeding frequency and food throughput. The San Juan teeth are not wearing fast. They are wearing slowly, in a manner consistent with infrequent episodic feeding, rather than the sustained daily consumption that fuels an animal running a high metabolic rate. Think about what that means in combination with the growth suppression documented in the bone histology. Lower metabolic demand means lower caloric requirement. Lower caloric requirement means the animal does not need to feed as often. Infrequent, opportunistic feeding on low-quality organic matter becomes viable precisely because the animal's physiology has been tuned to require less. This is an internally coherent survival system. Each adaptation reinforces the others. But the nitrogen isotope data introduces a complication that prevents the dietary picture from being too neat. Delta 15 nitrogen values in animal tissues track trophic position because nitrogen isotopes fractionate predictably as organic matter moves up food chains, with consumers becoming progressively enriched in the heavier isotope relative to their food sources. High delta 15 nitrogen values indicate high trophic position, meaning vertebrate prey. Low values indicate consumption of material near the base of the food web. The nitrogen data from the San Juan teeth does not produce a stable signal. It oscillates. Serial sampling along individual teeth shows intervals of relatively elevated delta 15 nitrogen values consistent with periodic consumption of vertebrate protein, followed by intervals of pronounced depletion that fall into ranges more consistent with fungal or decomposing plant material ingestion. What that oscillation describes is not a specialist, it describes an opportunist. An animal consuming whatever organic matter was energetically accessible, shifting between scavenged vertebrate carcasses when they were available, and lower trophic level detritivore resources when they were not. The olfactory hypertrophy documented in the endocranial reconstruction provides the sensory mechanism for exactly this strategy. an animal capable of detecting chemical gradients from decomposing organic matter across significant distances, navigating toward concentrated food sources in a landscape where food was scattered, unpredictable, and sporadically distributed, would have been well equipped for precisely the kind of dietary opportunism the nitrogen record documents. Here is the question the nitrogen oscillations make unavoidable. The intervals of elevated vertebrate protein consumption that appear in the nitrogen record require that vertebrate prey of some kind was available in the post-impact landscape. We know early Paleocene mammals were present at this site because their teeth came out of the surrounding matrix. Early crocodilians persisted through the KPG boundary. Early lizards and snakes survived. These small-bodied vertebrates would have represented the only significant mobile protein resource available to a large predator in a post-impact ecosystem. The nitrogen signal suggests this animal was catching or scavenging them opportunistically, not as a primary food source, but as a periodic supplement to a diet otherwise routed through decomposer pathways. Think about the energetics of that for a moment. A large-bodied terrestrial vertebrate relying primarily on fungal biomass, decomposing organic matter, and occasional small vertebrate scavenging. That is an extraordinarily low-quality dietary foundation for an animal of this size by any conventional metabolic standard. The viability of that strategy depends entirely on the metabolic suppression being as profound and as sustained as the bone histology indicates it was. If the growth ring data is telling the truth, and all the evidence suggests it is, this animal's caloric requirement may have been reduced to a small fraction of what a comparably sized Cretaceous theropod would have demanded. That fraction apparently was achievable in the impoverished post-impact ecosystem, barely intermittently and opportunistically, but achievable. Which forces the next question into focus immediately. Survival requires more than individual caloric balance. It requires reproduction, and reproduction requires more than one animal. The dietary record in the teeth of the San Juan specimen documents how a single individual managed to persist. But the broader question of how a lineage maintained demographic viability across 1.5 million years demands a completely different scale of evidence. Evidence that, as the investigation expanded beyond New Mexico, the rock in 2 other states was about to provide. The San Juan specimen stopped being a single anomaly the moment the second bone came out of the ground in Wyoming. That shift in scale matters more than almost anything else in this investigation. A single impossible fossil can, in principle, be explained away through mechanisms that feel uncomfortable but remain theoretically available. Unusual taphonomic preservation, undetected stratigraphic disturbance, some localized geological peculiarity that created conditions mimicking a Paleocene burial context in what was actually residual Cretaceous material. Paleontologists are professionally suspicious of single specimens precisely because the range of explanations for a single anomaly is always wider than the evidence can collapse. You can argue around one data point. You cannot argue around 3, geographically separated by hundreds of kilometers, independently dated, and displaying the same constellation of anatomical features. The Wyoming specimen came from the Hanna Basin, recovered from a road-cut exposure of the Fort Union Formation, which is the Paleocene terrestrial sequence that overlies the Laramie Formation equivalent across a wide swath of the Rocky Mountain Fallen Basin system. The Fort Union at this site had been mapped previously, sampled for pollen and mammalian microfossils, and assigned to the early Danian on the strength of its condylarth fauna and its position above a well-documented iridium-bearing boundary clay. What nobody had looked for, because nobody had any reason to look for it, was a large theropod. The bones were initially identified by a Highway Department geologist as unusually large fragmentary material of uncertain affinity, catalogued and held in a state survey collection for several years before the San Juan publication brought them to the attention of the research team and triggered a formal re-examination. The re-examination was unambiguous. 2 fragmentary hind limb elements, a partial tibia and a nearly complete metatarsal, preserved in Fort Union mudstone matrix, with cortical bone architecture that matched the San Juan femoral sections with a fidelity that bordered on redundant. The osteosclerotic densification was present. The reduced vascular porosity was present. The growth ring spacing was present at values implying annual somatic growth rates in the same suppressed range as the San Juan material. independent uranium-led dating of an associated volcanic ash layer placed the horizon at 65.6 million years ago, within the same early Danian window as the New Mexico material, and screenwashing of the surrounding matrix produced an identical ecological fingerprint: condylarth teeth, fern spores, and the fungal spore assemblages characteristic of post-impact disturbed substrates. The Colorado specimen arrived through a different pathway entirely: a private quarry operation in the Raton Basin, excavating Paleocene coal-bearing sediments from the Raton Formation, encountered an articulated partial forelimb, including a complete radius, ulna, and several carpals, preserved in a dark carbonaceous shale interbed within the coal sequence. The Raton Formation at this site spans the KPG boundary, and the position of the forelimb elements relative to the boundary clay, confirmed by both iridium geochemistry and a characteristic fern spike pollen horizon, placed them approximately 1.4 million years above the impact event. The forelimb bones were initially reported as belonging to a large crocodilian, which would have been stratigraphically unremarkable. But the osteological details of the carpals are definitively non-crocodilian. The arrangement of the carpal bones, their articular surface geometries, and the proportional relationships between the radius and ulna fall squarely within the morphological range of large-bodied coelurosaurs, and the cortical histology, once sections were cut from the radius, produced the same dense, low-porosity osteosclerotic tissue documented in New Mexico and Wyoming. 3 sites: Wyoming, Colorado, New Mexico, all independently dated to the same approximately 1.5 million year post-impact interval, all displaying the same unusual histological signatures. That is not coincidence. That is a population. And here is where the investigation crosses from paleontology into conservation biology, because the existence of a geographically distributed population raises a question that the fossil record alone cannot answer with any precision. How many animals are you actually dealing with? The minimum viable population concept, developed in the conservation biology literature to assess the demographic thresholds below which inbreeding depression, genetic drift, and stochastic demographic fluctuation make long-term persistence essentially impossible, gives you a framework for at least bounding the answer. For large-bodied terrestrial vertebrates with low reproductive rates, which the growth histology strongly implies this lineage had, the minimum effective breeding population size required to maintain genetic diversity and demographic stability, across intervals measured in the hundreds of thousands of years, falls in the range of several hundred individuals at an absolute minimum. Some population viability models, incorporating the full uncertainty around reproductive parameters for an extinct animal, push that floor considerably higher. Several hundred large-bodied theropods distributed across the western interior of North America throughout the early Paleocene. That is the implied population size, and the fossil record, before these 3 sites, contained exactly 0 evidence of their existence. Stay with the implications of that gap for a moment, because it is genuinely significant. The western interior Paleocene is not a poorly sampled interval. Formations like the Fort Union, the Nacimiento, and the Raton have been systematically prospected for vertebrate fossils for well over a century. The mammals of the early Paleocene are documented in extraordinary detail from dozens of localities across exactly the region where this theropod population was apparently living. Condylarths, multituberculates, archaic primates, early ungulates. The small vertebrate fauna of this time and place is known from thousands of individual specimens. If a population of large theropods was out there, it is reasonable to ask why it did not leave a proportional fossil footprint. The taphonomic bias argument partially addresses this. Large vertebrates are preserved as fossils at dramatically lower rates than small ones, for reasons that have nothing to do with their abundance in life. Large carcasses are disarticulated and dispersed by scavengers and hydraulic energy before burial can occur. Large bones weather rapidly when exposed at the surface. Large animals tend to occupy upland or well-drained habitats, where sedimentation rates are low and preservation potential is poor relative to floodplain and channel environments, where the small mammal record concentrates. The entire sampling architecture of the Paleocene fossil record is systematically biased against preserving large terrestrial vertebrates, particularly ones living in habitats where rapid deep burial was unlikely. That bias is real. But it does not entirely satisfy the question, and the research team was honest about that. Because you can use the same taphonomic bias argument to make almost any absence evidence disappear, and if you do that indiscriminately, you lose the ability to make meaningful inferences from what the fossil record does and does not contain. The more productive framing is to ask what the 3 sites that were found actually require in terms of population size and geographic distribution, and then ask whether the predicted population, given reasonable assumptions about taphonomic preservation rates for large terrestrial vertebrates in Paleocene depositional environments, is consistent with yielding exactly 3 known localities across more than a century of systematic collecting. The calculation is uncomfortable but not impossible. If you assume that large theropod carcasses in early Paleocene floodplain environments were preserved as recognizable fossils at rates comparable to those estimated for large Paleocene mammals from similar depositional contexts, and you back-calculate from 3 recovered specimens the minimum source population required to produce that fossil yield across 1.5 million years, the numbers do converge in the low hundreds to low thousands of individuals. That range is internally consistent with the minimum viable population thresholds. It is also consistent with the near-total absence of these animals from the rest of the fossil record. A population of that size, distributed across a landscape of continental scale, living at low densities dictated by the energy poverty of the post-impact ecosystem, would have been genuinely rare in any given depositional environment and would have left exactly the kind of sparse, geographically scattered, easily missed fossil record that the 3 specimens represent. The ghost population was real. It was viable, at least for a time, and its geographic distribution across 3 states implies that whatever allowed these animals to survive was not confined to a single refuge, but was a broadly operable biological strategy across the post-impact western interior. Which makes the question of why it ultimately failed all the more pointed. The population was not collapsing from rarity. It was not being picked off by competitors or predators. The mammal sharing its landscape was small-bodied and ecologically segregated from any meaningful competitive overlap with a large theropod. Whatever ended this lineage, it ended all of them, across hundreds of kilometres of landscape, within what the stratigraphic record suggests was an extraordinarily compressed interval. That kind of simultaneous, geographically coherent extinction signature points directly at one thing: a systemic environmental shift. And the teeth of these animals, read at sub-millimetre resolution, had been recording that shift 1 growth increment at a time. The teeth are not passive objects. Every living vertebrate grows its teeth incrementally, depositing enamel and dentine in layers that record the chemistry of the animal's body water, food sources, and ambient thermal environment at the precise moment each layer was mineralized. Those layers persist. They do not remodel. They do not exchange material with the burial environment at the rates that bone does. A tooth is essentially a sealed archive, and if you have the analytical resolution to read it at the scale of individual growth increments, you are not reconstructing paleoclimate from regional sedimentary averages or ocean core proxies. You are reading the biochemical diary of a specific animal, written 1 day at a time, in the mineral lattice of its own body. The serial isotopic sampling campaign conducted across the San Juan, Wyoming and Colorado specimens produced a dataset of that kind. Sub-millimetre resolution sampling along the growth axis of multiple shed teeth, each tooth representing a different growth interval in the animal's life, assembled into a composite temporal record spanning the individual lifetimes of several specimens. The analytical fidelity of this record exceeds what any sedimentary proxy from the same interval can provide, and what it shows is not simply a confirmation of the cold, impoverished post-impact environment already documented in the bone histology. It shows something far more consequential. It shows the world changing around these animals in real time, and it shows their bodies recording that change with a precision that makes the eventual outcome almost unbearable to read. Start with the earliest growth increments. The most basally formed enamel in the teeth of individuals estimated from bone histology to have been juveniles during the earliest portion of the sampled interval carries delta 18 oxygen values that, when converted to formation temperatures using the standard phosphate oxygen thermometry calibration for theropod body temperature ranges, indicate enamel mineralization at temperatures consistent with the profound post-impact cold, 4 to 7 degrees Celsius below the late Cretaceous regional baseline. That baseline itself is reconstructed from the same isotopic measurement applied to Maastrichtian theropod enamel from the Hell Creek equivalent beds below the boundary, so the comparison is internally calibrated and not dependent on an external paleoclimate assumption. These animals, in their earliest growth phases, were forming their teeth inside bodies that were operating at temperatures substantially below anything experienced by their Cretaceous predecessors. That is the world they were born into. That is the physiological baseline their developmental programs were set against. Now move along the tooth growth axis toward the later formed enamel, and then into the serially sampled teeth representing adult growth intervals. The delta-18 oxygen values do not stay flat. They rise. The shift is gradual at first, almost within measurement uncertainty across any single tooth, but cumulative across the full temporal span of the sample sequence. The directional trend is unambiguous. Approximately 2 to 4 degrees Celsius of warming per 100,000 years, sustained and progressive, embedded in the enamel of animals that were alive while it was happening. That rate and trajectory correspond with striking precision to the early Paleocene thermal recovery, documented in the deep-sea benthic foraminiferal oxygen isotope record from multiple ocean basins, a recovery driven by the gradual clearing of stratospheric aerosols, the resumption of volcanic greenhouse gas emissions from the Deccan traps, and the progressive rebuilding of biogenic carbon cycling as recovering ecosystems began processing and releasing organic carbon through decomposition and respiration. The teeth confirm that this global signal was reaching the continental interior of North America. The world was warming, not rapidly by human timescale standards, but persistently, directionally, and without reversal within the interval recorded by these specimens. The delta-13 carbon series tracks in parallel, and that parallel tracking is where the dietary and ecological dimension of the recovery enters the record. The earliest growth increments, corresponding to the period of most severe post-impact ecological impoverishment, carry the most isotopically depleted carbon values, consistent with the detritivore and fungal dietary base already established from the microwear and trophic level analysis. But as the delta-18 oxygen values climb toward warmer formation temperatures in the later growth increments, the delta-13 carbon values begin shifting as well, moving from the depleted range characteristic of decomposer-rooted carbon toward progressively more enriched values that isotope ecologists associate with the consumption of organisms feeding on photosynthetically fixed plant matter. The green food web is coming back, plants are recovering, herbivores are diversifying to exploit them, and the carbon chemistry being incorporated into these theropod teeth is picking up that recovery signal as the animals' diets gradually shifted to track whatever food sources the recovering ecosystem was making available. Stay with what that shift actually represents. This is not an animal eating exactly as it always had. The enamel chemistry documents a dietary transition in progress, a lineage that had survived on detritivore scavenging and fungal foraging, gradually incorporating more photosynthesis-derived carbon into its diet as the ecosystem around it rebuilt. That transition should represent improving nutritional conditions, more food, higher quality, more reliably available. For any normally physiologized vertebrate, the recovery signal in these teeth would be the signature of better times. Here is where the data becomes deeply strange. The stratigraphic position of the final theropod specimens at all three sites does not correspond to a period of ecological collapse or renewed environmental stress. It corresponds precisely to the interval in the delta-18 oxygen record, where the warming trend is most clearly expressed, and where the delta-13 carbon signal is most strongly shifted toward photosynthetically enriched values. The animals disappear from the sedimentary record not during the worst of the post-impact cold, not during the period of maximum ecological impoverishment, not during any interval that looks like deteriorating conditions by any conventional environmental metric. They disappear as conditions improve. The recovery is not background context to their extinction. The data places the recovery as the immediate temporal correlate of their disappearance. That coincidence demands a mechanism, and the oxygen record in the teeth provides the beginning of one. Atmospheric oxygen partial pressure is not directly recorded in tooth enamel oxygen isotopes, in the same way that temperature is. But the delta-18 oxygen values in tooth enamel do reflect the isotopic composition of the animal's body water, and body water composition in air-breathing vertebrates is influenced by the balance between oxygen intake through respiration and oxygen loss through metabolic water production. In animals whose respiratory physiology was functioning under hypoxic post-impact atmospheric conditions, the precise quantitative relationship between enamel oxygen isotopes and body temperature shifts in ways that are theoretically predictable, and that the research team modelled against the measured values. What that modelling produces, when applied to the terminal growth increments of the most recently formed enamel in the final teeth across all 3 specimens, is a signal consistent not with normal respiratory function at elevated ambient temperatures, but with a body struggling to maintain thermal equilibrium under rising heat load while simultaneously failing to extract sufficient oxygen from an atmosphere whose oxygen partial pressure was increasing faster than the animal's pulmonary architecture could accommodate. Think carefully about what that means. Rising temperature and rising atmospheric oxygen are both consequences of ecosystem recovery. Both should be physiologically neutral or beneficial for a normally endothermic vertebrate. For an animal whose thermoregulatory and respiratory systems had been restructured over dozens of generations to operate under chronic cold and reduced atmospheric oxygen, both of those changes were potentially lethal stressors. The enamel isotopes cannot prove that conclusion alone. They are a necessary but not sufficient line of evidence. The proof requires the bone tissue of the animal's final years, which tells that part of the story in an entirely different register. The periosteal bone deposited during the last growth intervals recorded in the most recently formed outer cortex of all 3 specimens contains something that the earlier growth rings do not: pathological remodelling. Not the uniform, cyclically banded tissue of a physiologically stable animal moving through predictable seasonal cycles, but disrupted irregular bone deposition with compressed and abnormally fused arrest lines, vascular channels showing erratic branching patterns inconsistent with normal metabolic throughput, and in the outermost periosteal layers, zones of apparent growth arrest that do not correspond to seasonal cold periods based on the isotopic temperature record. These are not winter lines, they are stress lines deposited during warm intervals, forming in the bone tissue of animals whose skeletal physiology was responding to a chronic sustained insult rather than a predictable environmental cycle. The bones were trying to keep building and kept stopping. The reason those stops were happening during the warmest intervals of the isotopic record and not during the coldest is the question that the next layer of evidence is going to answer with an anatomical specificity that goes well beyond what any isotope series can achieve on its own. The respiratory turbinals are the detail that changes everything. Most people who have followed theropod paleontology closely have never heard of them, because they almost never preserve. Turbinate bones, or more precisely the scroll-like cartilaginous and bony projections that line the nasal cavity in most amniotes, are among the most delicate structures in the vertebrate skull. They are thin, extensively perforated, highly vascularized, and entirely unsupported by the robust architecture that allows teeth and limb bones to survive millions of years of diagenesis and compressive burial. In the entire documented theropod fossil record, meaningful turbinal preservation is essentially non-existent, which is why the fragment recovered from the nasal cavity of the San Juan specimen, a partial maxilloturbinal element roughly 3 centimeters in its longest preserved dimension, represents something genuinely extraordinary entirely apart from any interpretive claim made about it. The fragment survived because it was buried within the nasal cavity itself, shielded by the surrounding facial bones from the compressive forces that flatten and destroy exposed cranial structures. It is incomplete, and the reconstruction of its original geometry requires careful comparison with the surrounding nasal passage dimensions. But the preserved portion is sufficient to establish 2 critical morphological features that drive the physiological interpretation of this entire lineage. First, the surface area. Respiratory turbinals in living vertebrates serve primarily as countercurrent heat and moisture exchangers, thin surfaces densely lined with mucosa across which inspired air is warmed and humidified before reaching the lungs, and expired air is cooled and dehydrated before leaving the body. The efficiency of this exchange scales directly with surface area relative to the cross-sectional diameter of the nasal passage. In most squamate reptiles, which do not rely heavily on turbinal function for thermoregulation, the ratio of turbinal surface area to nasal passage cross-section is modest. In endothermic birds and mammals, where respiratory heat and water conservation is physiologically critical, that ratio is substantially higher because the metabolic cost of losing warm, moist air with every exhalation is significant enough to have driven selection for expanded exchange surfaces. The San Juan turbinal fragment, when its preserved scrolling geometry is reconstructed using the available nasal cavity dimensions as a boundary constraint, implies a surface area to nasal passage ratio that falls well within the avian and mammalian range rather than the reptilian range. This is not a passive airway, this is an active exchange system. Second, the vascular architecture. The preserved fragment carries a dense network of foramina, small perforations through the turbinal surface that transmitted blood vessels supplying the mucosa. The density and diameter distribution of these foramina indicate a capillary network substantially richer than what would be needed simply for mucosal tissue maintenance. This is a heat exchanger designed for high blood flow throughput, capable of transferring significant thermal energy between the incoming and outgoing airstreams on every respiratory cycle. Biomechanical modelling of this reconstructed exchange system run against atmospheric parameters estimated for the post-impact environment specifically reduced ambient temperature and the somewhat different humidity gradients associated with a colder, more aerosol-laden atmosphere produces a striking result. The modelled heat recovery efficiency of this turbinal architecture under post-impact cold conditions exceeds 90%, meaning that roughly 9/10 of the heat energy added to inspired air during inhalation was being recovered from expired air before it left the nasal passage. For an animal running a suppressed metabolic rate in chronic cold, this level of heat conservation would have been the difference between thermal viability and inexorable heat loss. Without it, the metabolic cost of simply breathing in cold air would have drained energy reserves faster than the detritive diet could replenish them. With it, respiration itself became a heat-neutral or near-neutral energetic process, and the metabolic suppression documented in the bone histology became sustainable rather than ultimately fatal. Here is where the physiology starts locking into a single integrated picture. The trace element mapping of the cortical bone matrix, conducted across all three specimens using laser ablation, inductively coupled plasma mass spectrometry, provides the metabolic dimension that bridges the structural bone data and the turbinal function. Strontium to calcium ratios in vertebrate bone tissue are sensitive proxies for renal filtration efficiency because strontium, a calcium analogue, is discriminated against during intestinal absorption and renal reabsorption in ways that track the energetic investment the kidneys are making in mineral processing. High strontium to calcium ratios in bone indicate suppressed renal discrimination, meaning the kidneys are not working hard to exclude dietary strontium from the skeletal mineral pool. That pattern is documented in animals with chronically reduced metabolic rates, animals in deep hibernation, animals with sustained torpor states, and animals in ecological contexts where energy conservation has been pushed to physiological extremes. The San Juan, Wyoming, and Colorado specimens all show strontium to calcium ratios in their periosteal bone layers that are significantly elevated above the values measured in comparative Cretaceous theropod material from equivalent skeletal positions. The elevation is consistent and reproducible across the three geographically separated individuals, ruling out localized diagenetic contamination as an explanation. It reflects, instead, a genuine biological signal. These animals were running their kidneys at reduced output. Their hepatic metabolic throughput, inferred from rare earth element distribution patterns in the periosteal tissue that track the liver's role in regulating mineral homeostasis, shows corresponding suppression. The geochemistry of their bones is the geochemistry of organisms whose entire metabolic machinery has been dialed down to minimum operational levels. Not pathologically shut down, functioning, but functioning at a sustained reduced rate that has no parallel in any documented Cretaceous theropod, and that finds its closest analogues in the physiological states of large-bodied mammals entering prolonged winter dormancy or large reptiles in thermally imposed metabolic suppression. Except those analogues are seasonal and reversible, the trace element signature in the San Juan bone is not seasonal. It is present in the growth rings deposited during the warmest intervals of each annual cycle, not just the cold season arrest lines. This animal was maintaining suppressed metabolism year-round, not cycling through periods of elevated activity as ambient temperatures permitted. That distinction matters enormously when you start asking the question the research team was circling by the time the trace element dataset was complete. The debate over whether these physiological modifications were genetically fixed or facultatively reversible is not abstract. It has direct implications for why the animals died when conditions improved. A lineage retaining the genetic capacity to upregulate endothermy, to restore normal renal and hepatic throughput, to accelerate metabolic rate in response to environmental warming, would have been capable of physiologically tracking the early Paleocene thermal recovery. Warming temperatures would have triggered developmental or neuroendocrine responses that shifted the organism back toward higher output physiology. The turbinal architecture would have remained functional at reduced exchange demands. The osteosclerotic bone, once deposited, could not be undone, but normal remodeling processes could have gradually restored some vascular porosity over subsequent generations, as selection pressure for dense cold-adapted bone relaxed. The physiological lock-in would have been incomplete, and the lineage might have navigated the recovery. But 1.5 million years of sustained selection pressure for cold-adapted, hypoxia-tolerant, metabolically suppressed physiology in an isolated continental population creates a different genetic landscape than a single generation of phenotypic stress response. The critical question is whether the genetic variation necessary for high metabolic rate endothermy had been purged from the population through natural selection and genetic drift over the hundreds of thousands of years that the post-impact conditions persisted. Every individual in each generation that expressed higher metabolic rates, greater respiratory oxygen demand, and greater thermogenic capacity would have been at a selective disadvantage in a cold, hypoxic, food-impoverished environment. Those variants would have been filtered out. Generation after generation, the population would have become progressively more uniformly constituted for low-output cold physiology, not because any individual chose it, but because the environment relentlessly culled anything that deviated from it. By the time the climate began its sustained recovery, the genetic toolkit required to respond to warming, higher oxygen, and recovering food availability may have simply been absent from the population. Not reduced, absent, purged over dozens of thousands of generations by the most efficient selection environment imaginable, one in which the penalty for running a warm, metabolically active body was starvation and death, and in which that penalty was applied uniformly across an entire continent for over 1 million years. The turbinal architecture makes this irreversibility concrete in a way that genetic theory alone cannot. The nasal passage dimensions of the San Juan specimen, reconstructed from the preserved facial skeleton, are proportionally narrower relative to body mass than in any known large-bodied Cretaceous coelurosaur. The respiratory pathway had physically narrowed. The turbinal exchange system had expanded to fill that narrowed passage with maximum exchange surface. This is a system optimized for one specific atmospheric condition, cold and hypoxic, and its optimization required structural changes to the bony anatomy of the nasal passage itself. Bony anatomy does not reverse in response to environmental change within a single individual's lifetime. You cannot widen your own nasal passage as the air warms. The structural commitment was made at the developmental level, encoded in the growth trajectories of the facial bones, and it was permanent. Rising atmospheric oxygen partial pressure flowing through a system built for hypoxia is not simply suboptimal. It is mechanically mismatched in ways that biomechanical modelling of the reconstructed thoracic cavity and inferred air sac geometry makes quantitatively explicit. And that mismatch, playing out inside animals simultaneously being subjected to ambient temperatures their thermoregulatory physiology could no longer manage, is precisely what the final growth rings in the periosteal bone were recording, one disrupted layer at a time. The terminal signatures in those outermost bone layers are not ambiguous. They are the skeletal record of chronic physiological failure in organisms that had optimized themselves, over geological time, into a corner from which there was no biological exit. The pathological bone is where the mechanism becomes undeniable. Everything established about the metabolic suppression, the turbinal architecture, the trace element geochemistry, all of it describes a physiological system that had been refined over an extraordinary span of time into something highly specialized. Specialized systems work brilliantly inside the conditions that shaped them. Outside those conditions, the same precision that made them effective becomes the source of their failure. The outermost growth layers deposited in the periosteal bone of all three specimens document that failure in microscopic detail, and the documentation is consistent enough across three geographically separated animals that the mechanism cannot be attributed to individual pathology or local circumstance. Start with the arrest lines. Normal vertebrate bone, in a physiologically stable animal moving through predictable seasonal temperature cycles, deposits its lines of arrested growth during cold periods when ambient temperature drops, food availability decreases and metabolic rate falls in response. Those lines are thin, sharp and uniformly spaced relative to the seasonal periodicity that produced them. They are the bone equivalent of a clock. The growth rings in the middle and earlier cortical layers of all three specimens show exactly that regularity, tight cold season arrest lines, separated by bands of normally vascularized, actively deposited periosteal bone consistent with warm season anabolic activity. The clock is running correctly, but in the outermost layers, the most recently deposited tissue representing the final years of each animal's life, the clock breaks down. The arrest lines in those terminal layers are not thin. They are broad, irregular, and in several places multiple arrest lines are compressed together into zones of essentially static bone deposition that span what the isotopic temperature record in the surrounding enamel indicates were warm season growth intervals rather than cold season rest periods. These animals were stopping their bone growth during the warmest parts of the year. That is the inverse of normal. It means the thermal stress driving those growth arrests was not cold, it was heat. Look at the vascular disruption alongside those arrest lines and the picture sharpens further. The Haversian canals in normal periosteal bone run roughly parallel to the long axis of the element, branching at predictable angles and maintaining consistent diameters that reflect stable blood flow requirements through the tissue. In the terminal growth zones of the San Juan femur and the Wyoming tibia, the canal architecture becomes erratic. Branching angles deviate from the longitudinal axis at steep, irregular orientations. Canal diameters vary abruptly along their courses in patterns inconsistent with normal remodeling. Secondary osteons, the cylindrical packets of bone deposited around existing canals during remodeling, appear in abnormal densities concentrated precisely in the zones adjacent to the irregular arrest lines. This is bone tissue, responding to physiological disruption, attempting to repair and reorganize in response to a sustained insult that the normal remodeling program was not designed to manage. The specific character of that insult is what the isotopic record in the teeth, combined with the biomechanical modeling of the respiratory system, constrains to a precise mechanism rather than a general environmental stress. Rising ambient temperature alone is a plausible stressor for an animal whose thermoregulatory physiology had been calibrated for chronic cold. But large-bodied vertebrates have physiological buffers against moderate temperature increases, and the degree of warming implied by the delta-18 oxygen enamel series, while sustained and directional, was not rapid enough on geological timescales to constitute an acute thermal shock. The additional factor, and the one that makes the biomechanical modelling so critical to the overall synthesis, is the oxygen partial pressure increase associated with the recovering atmosphere. Here is why that matters in practical physiological terms. The reconstructed thoracic cavity geometry of the San Juan specimen, derived from the rib curvature, vertebral articulation angles, and the preserved sternal elements, implies a lung and air sac complex that had been architecturally reduced relative to the pneumatic systems documented in large Cretaceous coelurosaurs. The air sac system in advanced theropods functions not simply as a passive reservoir, but as a unidirectional flow-through ventilation mechanism that extracts oxygen from inspired air with substantially greater efficiency than the tidal breathing used by most non-avian vertebrates. The degree of pneumatization in the axial skeleton, which correlates with air sac volume and investment, is measurably lower in the San Juan specimen than in comparably sized Cretaceous material. The system was there, it was just smaller, less elaborately developed, apparently scaled back over generations in which atmospheric oxygen delivery demands were lower because the atmosphere itself was delivering less. An air sac system dimensioned for hypoxic post-impact atmospheric conditions, now receiving air with progressively higher oxygen partial pressure as the atmosphere recovered, faces a physiological problem that is not immediately obvious but becomes mechanically real under sustained conditions. Oxygen toxicity in vertebrate tissues is a genuine biological phenomenon documented in living animals subjected to elevated oxygen environments, producing reactive oxygen species that damage cellular components faster than antioxidant systems can neutralize them. The threshold at which this becomes physiologically significant is not the extreme hyperoxic conditions of laboratory oxygen toxicity experiments. It occurs at more modest elevations of oxygen partial pressure in animals whose antioxidant enzyme systems had been calibrated over many generations for lower baseline oxygen exposure. An animal whose biochemistry had been tuned for hypoxic conditions for over 1 million years of sustained selection pressure would not have the antioxidant reserve capacity of an animal evolved in a normally oxygenated atmosphere. Rising oxygen would have been, at the cellular level, progressively corrosive in a way that no behavioural or physiological adjustment available to these animals could have counteracted. Stay with the thermal component because it interacts with the oxygen problem rather than operating independently of it. Metabolic heat generation in a large-bodied vertebrate increases as ambient temperature rises because the animal must expend energy to maintain thermal homeostasis. For a normally endothermic animal with intact thermogenic capacity, that increase in metabolic heat production is managed through behavioural thermoregulation, panting, postural adjustment, and vascular shunting. For an animal whose thermogenic capacity had been suppressed to the degree documented in the San Juan bone histology, whose metabolic rate was running at a fraction of the expected value for its body mass, rising ambient temperature created a heat load that the reduced metabolic machinery could not dissipate through conventional endothermic mechanisms. The countercurrent heat exchanger in the nasal passage that had been such an effective cold weather adaptation now operated as a physiological liability, retaining heat in an animal that needed desperately to shed it rather than conserve it. The combination is what the terminal bone pathology reflects: hypothermia stress during warm intervals driving growth arrest, cellular oxidative damage from rising atmospheric oxygen, challenging tissues whose antioxidant systems were calibrated for lower oxygen exposure. The result is not catastrophic acute death. It is chronic, sustained physiological deterioration, an organism running outside the operational parameters of its own biology, month by month, year by year, unable to adapt because the genetic variation that might have allowed adaptation had been purged over 1 million years of selection for the opposite condition. Now consider the geographic simultaneity. The Wyoming tibia, the Colorado radius, the San Juan femur. 3 animals from 3 separate localities, each showing the same terminal pathological signatures in their outermost bone layers, each disappearing from the stratigraphic record within what the isotopic series constrains to less than 50,000 years of each other. That is geologically instantaneous for a population distributed across hundreds of kilometers of landscape. Stochastic local extinction, the kind of random disappearance that removes a small population from one site through disease, localized drought, or chance demographic collapse, does not produce synchronized disappearance across that spatial scale. What produces synchronized disappearance across a continental region is a systemic environmental shift that crosses every threshold simultaneously. The early Paleocene thermal recovery was that shift, not because it was rapid by any conventional definition of geological rapidity, but because the physiological thresholds of this particular lineage had been repositioned over an immense span of selective time so far from those of any other surviving vertebrate that what the rest of the Paleocene fauna experienced as benign normality was, for these animals, a crossing from tolerable to intolerable. The condylarths sharing their landscape were thriving in the warming world, the early birds were diversifying, the crocodilians were expanding their ranges as temperatures climbed toward their preferred thermal optima. The ecological recovery that spelled opportunity for every other surviving lineage spelled physiological catastrophe for the only one that had paid the full cost of surviving the worst of it. 50,000 years or less from the first clear appearance of the terminal pathological signatures in the bone to the last specimen in the stratigraphic column. That window is not long enough for evolutionary rescue, the process by which adaptive genetic variation arising through mutation can save a population from an environmental shift that its current genetic constitution cannot tolerate. Evolutionary rescue requires standing genetic variation or sufficiently rapid mutation to produce heritable adaptive responses faster than demographic collapse proceeds. In a population already reduced by 1.5 million years of ecological marginalization, already genetically impoverished through the sustained selection and drift documented across the histological record, the mutation supply rate and the generation time of large-bodied animals combined to make rescue mathematically impossible within the timescale the fossils allow. The last non-avian dinosaur did not die in the dark. It died in the light, in the warmth, in the oxygen, in the very conditions that every other creature on the recovering Earth was built to flourish within. The peristyle bone recorded the end of it, one disrupted growth layer at a time, the final biological accounting of a lineage that had survived everything the KPG boundary could deliver, and was ultimately destroyed by the world healing itself. What the complete synthesis of all this evidence confirms, and where the resolution of the Paleocene anomaly ultimately lands, is a conclusion that reframes not just this extinction, but the entire conceptual architecture of how paleontology thinks about survival, adaptation, and the costs of both. The evidence resolves. That is the phrase scientists use when a dataset stops being a collection of competing signals and becomes a single coherent story. After years of stratigraphic documentation, histological sectioning, isotopic sampling, biomechanical modelling, and geochemical analysis across 3 sites spanning hundreds of kilometres of the post-impact western interior, the Paleocene anomaly resolves. And what it resolves into is something that the discipline of vertebrate paleontology did not have a conceptual framework for before this investigation produced one. Let me walk you through exactly what the synthesis confirms, because the individual lines of evidence are compelling in isolation, but the full picture only emerges when they are read together. The theropod lineage that produced the San Juan, Wyoming, and Colorado specimens did not survive the Chicxulub impact through any single mechanism. There was no magic refuge, no singular physiological trump card, no geographic accident that sheltered a handful of individuals while everything else perished. What the combined record documents is a cascade of mutually reinforcing adaptations, each one building on the others, collectively constituting a survival strategy that was simultaneously brilliant and terminal. Start with geography, because it almost certainly mattered in ways the fossil record cannot fully quantify, but which population modelling makes theoretically necessary. The western interior of North America during the latest Cretaceous was a geographically complex landscape of basins, uplands, river systems, and varied depositional environments that would have created patchworks of microclimate heterogeneity. Some portions of that landscape would have been more buffered against the immediate thermal pulse of the impact, more sheltered from the early wildfire devastation, and more rapidly colonised by the fungal and decomposer communities that constituted the first viable post-impact food web. Whatever subpopulation of this Coelurosaurian lineage found itself in those buffered zones during the critical early post-impact interval would have had a marginally higher probability of surviving the initial mortality event. Marginal probability compounded across the hundreds of individuals that the population viability modelling requires is sufficient to establish a founder population. The survival at this stage was not heroic, it was statistical. From that founder population, selection began its work immediately. The cold was relentless and sustained. The darkness was chronic. The photosynthetic food web was functionally gone. Every individual in every generation faced the same pressures: reduced ambient temperature, reduced atmospheric oxygen, and a dietary landscape dominated almost entirely by decomposer cycle resources. The individuals whose bone histology happened to trend toward greater density, whose developmental programs happened to produce more metabolically conservative growth trajectories, whose olfactory systems happened to invest more neurological resources in chemosensory processing, those individuals survived preferentially. Those variants reproduced. Those traits accumulated in the population. Over hundreds of thousands of years, that accumulation produced the animal documented in the fossil record. Not through any single dramatic transformation, but through the slow, grinding work of differential survival applied generation after generation across a population already small enough that genetic drift was amplifying the directional selection signal rather than diffusing it. The osteosclerosis was not a mutation that appeared fully formed. It was the endpoint of a long gradient of skeletal densification that was progressively more favoured the longer the cold persisted. The olfactory hypertrophy was not an invention. It was the preserved terminal expression of a selection trend that had been running for longer than the entirety of recognisable human prehistory. The metabolic suppression was not pathology. It was the organism running the only physiological program that worked in the world it actually inhabited. Here is what makes this resolution genuinely significant beyond the particulars of this single lineage. The San Juan theropod and its relatives were not Cretaceous animals living unchanged in the Paleocene. The histology proves that definitively. They were organisms that had been genuinely transformed at the level of skeletal architecture, neural organization, metabolic programming, and respiratory anatomy by more than 1 million years of sustained directional selection. They were, in every meaningful biological sense, new organisms, post-apocalyptic organisms, animals built specifically for a world that no longer existed by the time they were the forms they had become. That is the core of the paradox and the core of the tragedy. The transformation that allowed survival was not reversible because it was not superficial. It went all the way down into the geometry of the nasal bones, the density of the cortical matrix, the proportional investment of the brain in different sensory modalities, the calibration of the antioxidant enzyme systems to lower atmospheric oxygen, the dimensioning of the air sac complex for hypoxic ventilation efficiency. These were not behavioral adjustments. They were structural commitments made by developmental biology, encoded into the body plan of every individual born after the selection gradient had done its work, permanent in the way that anatomy is permanent. When the climate began its sustained recovery, those structural commitments became physiological liabilities faster than any evolutionary process could convert them back into assets. Rising ambient temperature stressed thermoregulatory systems that had been optimized for cold retention rather than heat dissipation. Rising atmospheric oxygen challenged cellular antioxidant defenses calibrated for hypoxic baseline conditions. A recovering photosynthetic food web, which should have represented a return of dietary abundance, instead presented metabolic challenges to digestive and hepatic systems that had been running at suppressed throughput for so long that upregulating them required genetic variation the population no longer possessed. Think about what it means to succeed so completely at one mode of existence that you lose the capacity for any other. That is what the terminal peristyle pathology documents: animal by animal, growth ring by disrupted growth ring, not acute catastrophe. Chronic, incremental physiological mismatch. A body continuously trying to compensate for an environment that was drifting, degree by degree, oxygen increment by oxygen increment, away from the only conditions that body was built to manage. The synchronized disappearance across 3 sites within less than 50,000 years is the final confirmation that this was systemic rather than incidental. 50,000 years is a long time in human terms. In geological terms, it is essentially simultaneous. The same environmental shift crossing the same physiological thresholds in 3 geographically separated populations at the same moment means the threshold was real, the mechanism was biological, and the extinction was inevitable from the moment the climate began moving in the direction that every other surviving lineage was adapted to welcome. What this evidence collectively establishes rewrites a specific and important assumption that has structured paleontological thinking about the KPG extinction since the iridium anomaly was first identified. The assumption is that the extinction was caused by the impact. That is true in the most proximate sense for the overwhelming majority of non-avian dinosaur lineages. But the Paleocene Anomaly documents at least one lineage for which the impact itself was not the terminal cause. The impact was the pressure that shaped the survivors. The recovery was what finished them. The asteroid made these animals into something that could not die in the dark, and the returning light killed them instead. Paleontology rarely produces a result this clean. Usually the record is fragmentary enough, the evidence ambiguous enough, the interpretive gaps wide enough that multiple competing explanations remain plausible indefinitely. The Paleocene anomaly does not leave that room. The stratigraphy is unambiguous. The histology is unambiguous. The isotopes tell a coherent and internally consistent story across multiple independent lines of measurement. The biomechanical modelling of the respiratory architecture produces predictions that the terminal bone pathology confirms. Every analytical approach, applied to materials from 3 independent geographic localities, converges on the same mechanism. The last non-avian dinosaurs were not killed by the asteroid winter they survived. They were killed by the end of it. And somewhere in the Paleocene mudstones of the western interior, in basins that have not yet been systematically prospected, in Danian stage deposits that have never been screenwashed for large vertebrate material, there are almost certainly more bones, more growth rings, more disrupted periosteal tissue recording the final years of animals that paid an extraordinary biological price to persist in a broken world, only to find that the fixed world was something their transformed bodies could not survive. The investigation documented here is not a conclusion, it is a starting point. The ghost population left traces. The work now is to find them.